we're going to move to our next speaker, Dr. Anthony Norsha, who is a world expert in psychology. He's a professor of psychology at Stanford University, and he's done extensive research on behavioral, oculomotor, and electrophysiological functions in humans. And we'll answer any questions from Dr. Dubra's talk after Dr. Norsha finishes his. Dr. Norsha? Dr. Norsha, you might still be on mute. If you could unmute yourself.
Can you hear me?
Yes, we can. Thank you so much for joining us.
Okay, now I need to find my slides. Okay, great. So thanks for the opportunity to speak here. Really enjoying the sessions, learning a lot. I have no disclosures, and we've been supported by Glaucoma Research Foundation, National Eye Institute, and Research to Prevent Blindness. I'd also like to thank Jeff Goldberg for getting my group involved in optic nerve disease research and introducing me to Alf Dubra. And Alexandra Yakovleva has been very instrumental in collecting and analyzing data. Vladimir Vildovsky is central to our instrumentation development, which we do a lot of. And Naz Jahangir has recently joined the project, and she's helping collect data and doing some of our clinical informatics. So I will be talking specifically about the visually evoked potential. So Heather gave you a brief introduction to it. I'll drill down a little bit more into it and how it can reflect optic nerve disease. And in doing that, I'll talk about some of the advantages it has for this kind of work and some of the disadvantages as well. So as Heather was saying, clinical measures of visual function are typically subjective. Visual acuity is subjective. Visual field testing is subjective, and it's also something that a lot of patients don't like doing. On the other hand, it's very useful in that it gives us indicators of regional loss of function. So testing different parts of the visual field, producing a map of visual loss. And this can be then broken down into various sectors of the visual field, and there's some nice ways of connecting the sectoral functional losses to the anatomy. And so we're trying to move towards this with an objective measurement based on EEG. And the nice thing about EEG is a direct neural estimate, and it's objective. Also, it can be done in children of any age. This will be particularly useful for apples to apples comparisons that we heard Shannon talk about, and longitudinal studies. So EEG is really the oldest neuroscience measurement. This is a picture of Hans Berger in 1927, who was the first person to measure the EEG. And the picture here is his daughter Ilse with her eyes closed. And you can see this prominent alpha rhythm. And here she's calculating a sum. So there was a notion that the EEG was highly reflective of cognitive activity. We're more interested in using it as a readout for the output of the retina. So the VEP is based on the EEG. It's a specific response to sensory stimuli, visual stimuli in particular, and it's the activity that's generated in sync with changes of the visual stimulus. And what's useful for present purposes is that the VEP stimulus and analysis can be changed and differentiated and used to study different parts of the visual pathway. So if we have a optic nerve disease, we're really interested in function here, we'll be reading it out further along the pathway here after it's transited the optic nerve and the optic radiations to cortex. Somewhat indirect measurement, but nonetheless useful. And the other thing, which we haven't really talked about, is a lot of visual processing is done by cortex. And really, we have no idea about how cortex adapts to these deafferenting events in the eye. And there may be compensatory mechanisms that we would need to understand and have to make a cortical measurement in order to understand them. So VEP measurement of visual function, just a brief summary of how VEPs are generated. So this is where the electrodes as red dots are located with respect to the brain. And we can see the cortical gray matter, which is the key structure that generates the VEP. So the gray matter is where the EEG comes from, not the white matter. And the standing model of how VEPs are generated is they're generated by these elongated pyramidal cells. They're called pyramidal because their cell body looks like a pyramid. So they're oriented at right angles with respect to the cortical surface. So they have an orientation which is critical because as synaptic activity dumps current into these cells, it flows in a elongated direction. And this allows for what's called a far field to be generated. So there's a current flowing in the cell. This generates a radiated field that we can then pick up on the sensor array here. And so this is an imaging modality where we're taking the measurements here on the sensors and tracing them back to where they were generated in cortex. So this is a VEP from early visual cortex. So this is the readout that we'll be using to be looking at optic nerve function. And like clinical visual field testing, we're also interested in moving this technique towards a way of measuring visual field loss in sectors, and we're just making our first baby steps in that direction. And so we're starting off by using arrays of small points of light, and we'll eventually be moving into modulating these in sectors. Right now, the data I'll show you today came from the whole field. We're at the point of being able to record the lower visual field and the upper visual field at the same time, but I won't be talking about that. So these arrays will have little points of light in them that are flickering. These are brightening points, and we'll also use darkening points later on in some of the work on our glaucoma patients. So I'll illustrate what we're doing with glaucoma as an optic nerve disease, and we hope to be able to make exactly these same measurements in patients with optic dystrusion. And one of the nice things that we'll be able to do is we'll be able to use the same control participants. So I'll show you data from 36 eyes of healthy age-matched controls and 54 eyes of patients with glaucoma. And so what we'll do with the glaucoma patients is we'll split them into two groups on the median of their visual field deviation. So it happens that when you split them that way, the field loss, the mean deviation, splits at about -6 dBs, which is a kind of a typical breakpoint in ophthalmology, clinical ophthalmology, for mild versus moderate levels of visual field loss. So first data we'll look at is VEPs from controls versus patients with moderate glaucoma. And so spend a minute on this. So we're looking at voltage as a function of time. And right at time zero, there is a rapid change in the stimulus. And that rapid change in the stimulus is what we're time locking the analysis to. And then we can average the data to improve its signal to noise ratio. So we'll see voltage as a function of time. Nothing happens for, say, 50, 60 milliseconds. And then that's the signal getting through the retina and up the optic nerve and into cortex. And then finally, cortex is able to receive this input and process it. And what we see here is the control deflection is about 3 times larger in amplitude than the deflection measured in the patients with moderate glaucoma. Now, it's been known for many, many years that the VEP is quite sensitive to glaucoma, so this is not at all surprising. And so what we have here is exactly the same measurement made in the other half of the eyes with glaucoma, the ones with milder glaucoma. And we can see there's also a daylight here between the patients and the controls, but not quite as much. So if we put all 3 of them together, we can see there's a nice grading of the loss of amplitude. And there's a very subtle slanting of these curves. Also, the response gets slower as the amount of field loss gets deeper. So that's another parameter we can measure. We can have not only how big the response is, but how fast it is. Gives us a bit more information, which may be useful in the future. So this is not at all surprising. As I said, this sort of thing has been known for many years. And the thing that we've been interested in is probing a little bit deeper based on work that's come out recently in mouse models of glaucoma, where the mouse models, there are now about 4 or 5 papers on this, that show that the on and off visual pathways in the retina are differentially vulnerable to glaucoma. And in particular, the off pathway in the inner retina is damaged before the on pathway. And so we were interested in seeing whether this might be an early indicator, a differential loss between on and off pathway function might be an earlier indicator of loss in glaucoma. And the challenge then in humans is to be able to make a measurement that is associated with these anatomical divisions in the inner retina. And we are using a technique that's come out based on physiology of ganglion cells and a bunch of psychophysics, which says that you can preferentially stimulate the on pathway by presenting luminance increments and the off pathway by presenting luminance decrements. Now, this is not an isolation of the pathways, it's a biasing the response to the on pathway versus the off pathway, depending on whether the stimulus is rapidly brightening or rapidly dimming. And so that's what we'll show the participants, and we'll make separate measurements for increments and decrements. And so what we find is, like in the mouse models, that we get a better differentiation of glaucoma severity if we use the decremental responses than the incremental responses. So I'll just go through that here. So we split the data into the on responses. These are the mild and the moderate severity glaucoma patients. And here are the off responses. So there's a general smaller response to on than off in our data. But what's interesting to us is that there's very little difference between mild and moderate severity glaucoma. There's very little daylight here, but there's quite a bit more daylight here for the off. So we think it's potentially more sensitive as an indicator, may be able to differentiate different degrees of optic nerve functional loss better than increments. And so we're actively pursuing this now in glaucoma patients, and we've just seen our first 2 optic disc drusen patients. And so why we think this is maybe of interest in the case of optic dystrusion is in general, it would be useful to be able to make measurements of the same type at all ages, given that this disease does start in childhood or perhaps even in infancy. And so we can make the same measurement with the same measurement criterion. And then one of the questions that we're particularly interested in is the damage mechanism that we see in glaucoma present in optic dystrusion as well. That is the off pathway just generally vulnerable to any kind of disruption of optic nerve function? Or on the other hand, is this a particular pattern that's only seen in glaucoma? And I think it would be interesting to think about this in papilledema, which is something I wouldn't have thought of doing having not seen the presentations earlier today. So I think looking for these signs and whether the manifestations of functional loss differ between these will be very useful clinically and also in terms of understanding the pathophysiology of these different disorders. And we're particularly excited about making structure-function correlations. Alf Dubra and colleagues are developing some new visible light OCT techniques that have much better resolution. We'll be able to actually image the separate on and off ganglion cell letters in the inner plexiform layer, and then make very direct structure-function correlations between retinal layers and the cortex. And we have another project with a colleague, Joyce, has brought on board Justin Gardner in psychology, who's an expert in functional MRI. And here the idea would be to look at possible cortical reorganization that happens secondary to optic nerve disorders. And with that, I'll end and thank you and take your questions.
Thank you, Dr. Norsha. That was terrific talk. We have one question from Mr. Bradley Katz. He asks, are you successfully employing the multifocal VEP to study either glaucoma or drusen?
We're moving in that direction. We took inspiration from the multifocal VEP in the array format that we're using, which is scaling the elements to compensate for the cortical magnification factor. But at this point, we're not lighting up the individual hexagons like in a true multifocal design. We're moving gradually towards that, one step at a time. We're currently doing a lot of hemifields, so the upper half will be modulated separately from the lower half. We can read those out separately. Once we get that working well, we'll try to go to quarter fields, maybe even to eighths of fields. That's probably about the limit that we'll be able to do.